So good morning students. Botany assignment 26/8/2020. Coming to question number 1. Charging. Charging is nothing but amino acylation. Charging of tRNA involves the attachment of amino acid to tRNA. Amino acid to tRNA. Before that, amino acids gets activated by taking energy, by taking ATP, they gets activated and that activated amino acid transfers to third end of tRNA, that is CC end of tRNA. These two reactions were catalyzed by same enzyme. What is the name of that enzyme? Amino acyl synthetase enzyme. So right answer for this question is first option, amino acid to tRNA. Then coming to question number 2. What is the purpose of untranslated regions? UTRs present on mRNA. On the mRNA, this is start codon AUG and this is stop codon. This is UAA and these are the sense codons. All these are sense codons. All these are sense codons. This is stop codon. This is stop codon and this is start codon. So before the start codon, untranslated region is present. After the stop codon, untranslated region is present. Before the start codon, untranslated region is present. After the stop codon, untranslated regions are present. This is 5' and this is 3'. Here, guanosine eukaryotes. So guanosine triphosphate G cap is present. Here, poly A tail is present. Poly A tail. So these untranslated regions that are present before the start codon and after the stop codon, they helps in effective translation mechanism. For efficient translation, so answer for this question is third option. And then coming to question number 3, non-proteinaceous enzyme that acts as catalyst for the formation of peptide bond is non-proteinaceous enzyme. All enzymes are proteins except ribozyme. All enzymes are proteins except ribozyme. And this ribozyme is nothing but 23srRNA, which is nothing but polynucleotide. It is not polypeptide, it is a polynucleotide. So, right answer for this question is second option, that is ribozyme. And then coming to question number 4, activation of amino acid. Activation of amino acid during protein synthesis requires the participation of specific molecule of tRNA. Activation of amino acid during protein synthesis requires a participation of specific molecule of tRNA. First amino acid gets activated by taking ATP and also this reaction is catalyzed by amino acid synthetase. So amino acyl AMP enzyme complex is formed and this amino acyl AMP complex again it attaches to third end of tRNA, that is CC end of tRNA, so that CC end otherwise called amino acid binding site. So tRNA is the right answer. tRNA is the right answer. For question number 4, second option is right answer. And then coming to question number 5, during protein synthesis, a ribosome moves from end to end of mRNA to recognize all codons. This movement of ribosome, the relative movement of ribosome by 5' to 3' direction. The relative movement of ribosome on the mRNA from 5' to 3' direction is nothing but ribosome translocation. It is called translocation. Ribosome translocation. This requires an enzyme that is called translocase. Ribosome movement, relative movement of ribosome on the mRNA from 5' to 3' direction. Every time one codon distance is nothing but translocation. So, right answer for this question is second option, that is translocation. Then coming to question number 6. During protein synthesis, amino acid gets attached to tRNA with the help of amino acyl tRNA synthetase enzyme. Amino acyl tRNA synthetase enzyme plays dual role. First function is activation of amino acid. Second function is transfer of activated amino acid towards CC end of the tRNA. So, answer for this question is second option. And then coming to question number 7. The first amino acid in any polypeptide chain of prokaryotes is always protein synthesis begins with AUG. AUG codes for methionine in the case of eukaryotes, but whereas in prokaryotes, formyl group is present to the methionine, so formyl methionine. In the case of prokaryotes, first amino acid that binds to first amino acid in the protein molecule is formyl methionine. But in eukaryotes, methionine, formyl group is present to methionine in the case of prokaryotes. So right answer for this question is third option, that is formylated methionine. Then coming to question number 8. At which end of mRNA translation always begins? At which end of mRNA translation always begins? Fifth end. At which end of mRNA fifth end translation begins? 5' to 3' direction ribosomes binds to AUG, that is fifth end. Right answer for this question is second option, that is fifth end. The process of translation, next coming to question number 9. The process of translation requires transfer of genetic information from a polymer of nucleotides to a polymer of amino acids. Okay, that is what we call as translation. Polymer of nucleotides, that is codons present on the mRNA, to the polymer of amino acids. Polymer of amino acids, these are amino acids. In between amino acids, peptide bonds are formed. These are peptide bonds. So right answer for this question is second option, that is amino acids. Which type of nitrogen bases are present in loops of tRNA? Which type of nitrogen bases are present in bases of tRNA? You know, Holly proposed a cleverly model to explain the structure of tRNA. It shows 3 main loops. These are the 3 main loops. One accessory loop, and then one tail is present. One tail. This is 5' and this is 3'. In the tail, 3 unpaired nucleotides are present. This is amino acid binding site. 3 loops are present. This loop is anticodon loop. This is tail. The loop that is present opposite to tail, 3 nucleotides are present. They form into anticodon. Codons are present on mRNA. Anticodons are present on tRNA. During protein synthesis, codon and anticodon, they show complementary base pairing. And then these are the lateral loops. 2 lateral loops are present. This is lateral loop and this is another lateral loop. This is DHU loop. This is T-C loop. They contain these two. This requires enzyme recognition loop. Enzyme recognition loop. This loop recognizes ribosome. Ribosome recognition loop, and this is enzyme recognition loop. DHU loop recognizes enzyme, and this is anticodon loop, and this is T-C loop, ribosome. In these 2 loops, both usual and as well as unusual nitrogen bases, hydrouridine, and then T-C loop, uracil. So dihydrouracil. So both usual and unusual nitrogen bases are present. Both usual and as well as unusual nitrogen bases are present in the loops. So right answer for this question is third option, that is both usual and unusual nitrogen bases are present for question number 10. And then coming to question number 11, a group of 3 nitrogen bases on tRNA to recognize a codon and mRNA is called 3 nitrogen bases present on the tRNA to recognize codon. This is codon on the mRNA is what we call as anticodon. If anticodon contains UAC, codon AUG, AUG codon present on the mRNA, and then UAC anticodon present on the tRNA during protein synthesis. In between these 2, complementary base pairing occurs. Hydrogen bonds are formed. Anticodon. So right answer for this question is anticodon. Anticodons are present in tRNA, and then codons are present on mRNA. Then coming to question number 12. In actual structure, the tRNA is a compact molecule that looks like inverted L shape. This is a flower leaf model proposed by Holly, 2D structure. But in actual structure, the tRNA looks like inverted. The tRNA looks like inverted L shape. Inverted L shape. This is anticodon. and this is 5 dash, 3 dash, inverted L shape. In original position, it is inverted L shape. Original position, the tRNA is inverted L shape. Right answer for this question is second option. During protein synthesis, which one is code on mRNA is translated? That is right answer. Coding is done by DNA strands, wrong. And then code is transferred on tRNA, that is also wrong. All of these are correct. So, the correct answer for this question is first option. Code that is present on the mRNA is translated. As a result, protein molecule is formed. Okay, na? So, thank you, students. Thank you very much.